Happy New Year and welcome to our Grand Round series. Our Greenberg bladder cancer team is grateful you are here. Of course, seeing you in person would be preferable, but given the ongoing response to managing the SARS-CoV-2 pandemic at Hopkins and in our local communities, these virtual platforms have proven to be an efficient means for sharing information, for education and discussion. So first, a couple of updates. Next month, February 22nd, please join us for an engaging conversation with Dr. Nirmish Singla around kidney health and bladder cancer. For updated information about the Institute, be sure to simply Google Greenberg Bladder Cancer Institute. Further, I wanted to draw your attention once again to our evolving group of programs specifically tailored around the experiences of women with bladder cancer, developed and led by Drs. Armin Smith, Jeannie Hoffman-Sensis, and the program team of Pamela Getz, Samantha Rockler, and Stephanie Cooper-Greenberg. Their programming details are also available on our website. And of course, please never hesitate to reach out and we will send you the details directly. Several weeks ago, we activated a new section of our website focused on upper tract urothelial cancer, UTUC. Our UTUC leaders, Dr. Jeannie Hoffman-Sensis and Nirmish Singla, have outlined several new clinical trials and a designated email address to access more specific UTUC information. Of interest, a new newsletter will be out shortly, and we will share our important updates with you directly. Grateful to Stephanie Cooper-Greenberg for her leadership in developing this for us. Again, all of this information and more is up on our website. And if we can help you directly, we are only an email away at bladdercancer@jhmi.edu. I'm incredibly grateful to all of you for joining us today for an introductory conversation about the immune system and bladder cancer. We thought you might appreciate taking a little deeper dive into the biology of how the immune system affects bladder cancer, given the importance of immunotherapy and its treatment. This session can go in many different directions, and I encourage you to ask any and all questions. I hope you'll allow me the chance to provide a brief introduction of today's discussion leader. So Dr. Burles Rusty Johnson is an instructor of oncology at Johns Hopkins University. He did his undergraduate work at Augusta State University. He then moved to Medical College of Georgia, where he completed his MD, PhD, and residency training before joining us here at Johns Hopkins for his oncology fellowship. I am particularly grateful to Noah Hahn for convincing him to stay with us. In 2020, he was awarded the Bladder Cancer Advocacy Network's New Discoveries Young Investigator Award for his work, which is focused on understanding how B cells influence anti-bladder cancer immunity. In addition to his laboratory research, he has built a strong and growing clinical practice and serves as the point person for us and our collaborators for questions related to the immune system. We are thrilled to have Rusty join us today, and he has agreed to speak for about 35 to 40 minutes, and then we'll jump into a larger group discussion. And if it goes a little bit beyond the top of the hour, we can accommodate that. I would ask for folks to please submit questions via the chat feature. And after Rusty's overview, we will have ample time for group discussion. If there's a really burning question, let me know and I'll go ahead and interrupt him. So Rusty, over to you.
David, thank you very much. Let me go to my share screen and I'll put my, put it in, there we are. Okay, so everyone can see this. So thank you all so much, Dr. McConkey. Thank you for the wonderful introduction. Thank you all for having me here. So I thought it'd be great to discuss the immune system interactions with bladder cancer because we know about the immune therapies that are approved for bladder cancer and discuss the good and the bad side of immunology with regard to this. So just disclosures, I am on an advisory board for Seattle Genetics and then also co-author on a patent that we're looking at using immune-based gene signatures to predict outcomes in response to immune checkpoint inhibitor therapy. So for my talk, I wanted to talk about how the immune system helps us, how the immune system interacts with cancer, how understanding the immune system interactions with cancer help with treatment development. And to stimulate the immune system, I have a plus sign here, or inhibit the inhibitor, 2 minus signs here for putting the accelerator on, or maybe you've heard of taking off the brake. We'll discuss that as we go along, how modern immunotherapy for bladder cancer works, and also how understanding immunology can help predict patients who have the best chance of responding to immune therapy. So most of you, if not all of you, have had an infection before, and just to discuss briefly how the immune system clears that infection and how that can relate to bladder cancer treatment. So when you get an infection, you have this virus, and then a virus will infect cells here in green, and you have uninfected cells, and then this viral load will increase over time, and then it will decrease, and as the immune system clears the virus, and also other infections you could also extrapolate here. And that occurs as immunity increases. So as your immune system begins to be able to control and then eventually clear the infection, the immune system goes up, and then once the infection's cleared, once the immune system cleared the infection, then the immunity can go down. Forgive me for having the CD8 T cell here. I understand in virology or viral immunology, there's a lot of immune cells involved, but this is just to show you how an infection is typically cleared with or by the immune system. And that becomes important when we look at Coley's hypothesis. So William Coley was a surgeon, lived in the late 1800s and early 1900s. He's actually considered the father of immune therapy. And the reason he's considered that is because back in the 1880s, I believe, he had a young woman in late teens, early 20s who died of metastatic sarcoma after amputation. Now, we've come a long way in cancer therapy. Back then, amputation was the standard of care for patients who had sarcoma. So he went back and asked himself, well, can we improve treatment for these patients? And he did a chart review, and he found that a patient who had sarcoma actually developed tumor regression after developing a skin infection called erysipelas. I can't say it right, so forgive me if I'm mispronouncing it, because I don't call it that anymore. I call it cellulitis or a skin infection. In 1891, he started injecting patients, and actually up to over 1,000 patients he injected with over a period of time with these toxins called Coley's toxins. Actually, a concoction of a strep subtype and a serratia, for those of you who are interested, led to tumor regression in some patients, but also led to public criticism. Most of you know, Journal of American Medical Association is a fairly well-known journal, and he endured some very, very public and strong criticism. Now, his daughter actually launched a Cancer Research Institute, which is still going on and still funding junior faculty, postdoctoral fellows, and a number of people. And but in 1962, FDA declined to recognize Coley's toxins as proven drugs. It actually was illegal after that time to treat patients with Coley's toxin, despite its efficacy in some patients in the past. So how does this relate to bladder cancer therapy? Well, we're actually using a infectious subtype, a weakened non-harmful strain of Mycobacterium bovis to treat patients with non-muscle invasive bladder cancer. So we're using Coley's hypothesis to treat patients, not necessarily Coley's toxin, but Coley's hypothesis. So for those of you who don't know, I'm sure most of you do know that BCG is a vaccine for infants against tuberculosis where TB rates are high. That's the BCG vaccine. And then also the weakened strain of BCG that we use treats high-risk non-muscle invasive bladder cancer. And it's also the first FDA-approved immune therapy. So, and we'll talk about that as we go along. So on the right here is basically a historical overview of BCG. So go back to Coley's hypothesis and Coley's use of Coley's toxins, and then Robert Koch discovering Mycobacterium tuberculosis as a cause of organism of TB in 1882. Then Calment and Guerin, forgive my French pronunciations, but they began to develop the BCG vaccine. That's why it's called Bacillus Calment-Guerin. And then the BCG vaccine was developed. And then another thing that was important was Raymond Pearl found that patients with lesions of active TB had a lower incidence of cancer in postmortem studies. So these people who had active infection had a lower incidence of cancer when they died. Then the Lubeck disaster, where a number of neonates, 251, were actually orally given contaminated BCG. They actually developed tuberculosis, and then 72 actually died of TB, sadly. So kind of had a setback for this area of research. But more important, but also importantly, Lloyd Old, who's another well-known immunologist, reported that if you infected mice with BCG, those mice showed increased resistance to tumor cell transplantations. Give tumor cells, try to implant tumors into mice after BCG infection, and those mice, fewer of them developed and grew tumors. And then Alvaro Morales devised the original BCG protocol for intravesicular use, directing from Lloyd Old's research, and also going back to Coley, all the way back to Coley. And then the first use of BCG was done in 1976, and then BCG was approved in 1990. So again, going from infection and the importance of how the immune system interacts with infection to now using an infectious agent to treat patients with cancer. Huge milestone, but it took about 100 years. So do immune cells facilitate tumor elimination or tumor growth? The answer to that is really, it depends. So the immune cells function to eliminate tumors. So there's immunogenic tumor cells and non-immunogenic tumor cells. So our tumor cells basically have a lot of proteins on them that allow them to be recognized and eliminated by the immune system, or do they express proteins that say, don't attack me? And we'll talk about that as we go along. If they have the appropriate proteins on these tumor cells, then the immune cells can actually recognize them, attack them, and eliminate them. And that's called tumor surveillance. When you hear immunologists mention tumor surveillance, that's all they're talking about is your immune system is like the world's best security system, just going over and around to the body, in and throughout the body on a complete whole body level, and actually looking for anything, infectious agent, foreign body, whatever, tumor, and asking a question, do we attack or do we not attack? In this case, the immune system appropriately attacks. In this case, the immune system does not attack. So we'll talk about chronic inflammation in a minute, but tumor cells can express, again, proteins that say, don't attack me. And then the immune cells turn from your best friend to your worst enemy. And the immune cells actually facilitate the tumor progression. And then you end up with cancer that's, in this case, inflammation induced. Robert Schreiber, you'll hear his name as we go along. He's involved in cancer immunoediting, and we'll talk about what that is in a minute. So chronic inflammation, we talked about how that inflammation just a minute ago can actually encourage the tumor growth. And how does that occur? Well, environmental exposure. All of us know about Agent Orange and bladder cancer, or most of us do. So that can be an environmental cue that promotes cancer, that promotes the chronic inflammation. It causes it. Dietary exposure, obesity, is another risk factor. Diabetes, another risk factor for cancer. Anything that causes chronic inflammation. Again, chronic infection, chronic UTIs in women with bladder cancer. I know I heard this time and time again. And the reason that it's a risk factor for bladder cancer is because of the chronic inflammation that occurs that allows that immune system to be just activated enough to where it doesn't clear the tumor, but it allows a tumor to grow, that chronic inflammation. And then autoimmunity, I don't know if y'all are aware, but in patients who have pancreatic cancer, those patients who have autoimmune pancreatitis actually have a higher risk of a pancreatic cancer development. So you can have chronic inflammation, inflammation caused by obesity, diet. You can have therapy-induced inflammation, so you can get inflammation directly from therapy as well. Tumor itself can actually perpetuate. You know, we have snow going out right now in Maryland, or looking like maybe a snowstorm later this week. You know, it's a snowball effect. Once that tumor starts growing with that chronic inflammation, the tumor itself can propagate that inflammation and facilitate its growth. So Robert Schreiber, I mentioned him a minute ago. He came up about 20, 30 years ago with the 3 E's of cancer immunoediting. So this kind of gets at the question of how does cancer develop? Does it? So The immune system, when it interacts with a tumor, it can say, okay, well, we're getting rid of this thing. Major immune activation, and all those immune cells go in, they eliminate the tumor. That's what you want, and you want it to be eliminated before it becomes clinically relevant. That's what happens a lot of the time. Okay, we don't see it, we don't know it, it just happens. The immune system's great at that. What happens if a tumor becomes starts to grow, but it doesn't go everywhere? Okay, so we're right now talking about our non-muscle invasive bladder cancer. You have equilibrium. So that equilibrium is where the tumor, the immune system is allowed or keeps that cancer under a level of control, but the cancer still is alive. The cancer is still there. And the cancer eventually, if the cancer is not taken care of at this state through surgery or other means, the cancer eventually develops enough mutations and enough what we call escape variants, or ways of escaping the immune system control, to where that's when you end up, that's when a patient ends up with metastatic disease. Disease has gone to a liver or lung somewhere else, because it's no longer under local control, which is equilibrium, which is kind of your non-muscle invasive bladder cancer, or even your muscle invasive to some extent, where it's kind of going in equilibrium. The immune system starting to lose control when it's muscle invasive. This is a little more complicated, but I wanted to, for those of you who want to know about how this process works, for the purposes of this talk, we really don't need to know what types of cells these are. Just know that these cells that are colored are immune cells, and these cells that are in gray here are tumor cells. So in elimination phase, these immune cells are able to go and attack, kill off the cancer, get rid of it. In equilibrium phase, you've got some of these red cells here that are the tumor cells that are kind of starting to escape. They're getting a little more aggressive. The immune system's not able to control it as well. But right now it's under some sort of control. And then escape is when these tumor cells develop enough mutations and enough ways of escaping the immune system. One of my mentors mentioned the tumor as something that just keeps pushing a bunch of buttons on a typewriter. They keep pushing the different buttons on the typewriter to keep it, activating the different pathways until it's able to escape, unless something is done to get rid of it before, to get it back into the elimination phase. So escape is when you have, and a patient has metastatic disease, it's escaped immune system control. So treating bladder cancer, what to do? So, there's two different approaches really for immune treatment. You can either focus on stimulating the immune system, that's the plus sign, and forgive me for my math here, but or you can inhibit the inhibitor, which is, to me, is a double minus, because the inhibitor is immune inhibition. It's a minus sign. It's a minus sign on attacking that cancer, and then you inhibit it. So you put a minus on the minus sign, and then I know they say that two wrongs don't make a right, but in this case, two wrongs do make a right, because you end up with a minus, and then that minus, and then you end up with the immune activation, which is taking off the brake. So again, immune inhibition, that's one minus. The blockade of the immune inhibition, that's the other minus, and then you hopefully reestablish cancer control. And that the best example of that is, as you probably would already guess, are the immune checkpoint inhibitors in bladder cancer. Going back up to the top, immune stimulation, pushing that accelerator to try and, think of it as beating your head against a brick wall, which I'll show you some pictures of brick walls in a minute, explain why. But if you break that wall, if the wall is breakable, if it's not that tall, or if it's easy to break, then you can get through it by stimulating the immune system. Transfusion or injections of factors meant to stimulate the immune response. Best example of this is BCG and bladder cancer. And then the adenovirus mediated. So what is that? That's a virus that actually that's the interferon gene therapy. We can talk about interferon as we go along in this talk, but basically it's a gene therapy you can inject intravesically into the bladder, and you can actually cause immune expansion and then elimination of that cancer inside the bladder. And we're talking about non-muscle invasive cancer here. So immune stimulation, good for localized non-muscle invasive bladder cancer, inhibiting the inhibitor, better for your metastatic disease. And then there's therapeutic vaccines, Sipuleucel T, you may be aware of prostate cancer. And then you can actually take patient cells out and then expand them. Steve Rosenberg at the NIH has done this a lot, and then infuse them back into the patient. You know, we're not in prime time for that for bladder cancer yet, but that's something that might, you know, that's looking, you know, could be on the horizon. And then for your liquid tumors, your lymphomas, your hematologic malignancies right now, that they'll take the immune cells out, and then they'll genetically engineer them and then put them back in. So lots of ways to modulate the immune system. And I'm only talking about a few of them here. Lots of ways to modulate the immune system, lots of potentials for bladder cancer to take these approaches and harness them for bladder cancer in the future. So overcoming the wall of immune suppression. So I just wanted to, again, hone in on the different levels of immune suppression that can occur in a patient depending on what type of cancer they have with bladder cancer and how aggressive it is, and how that dictates what therapy that you end up getting. So in this case, non-muscle invasive bladder cancer, I was practicing for my talk earlier, and I said to myself, you know, my 7-year-old can get over this wall, right? I'm not saying it's that easy at all, but I still think I could probably walk over that wall. So in non-muscle invasive bladder cancer, the point I want to make is, yes, it's cancer, but the immune system still has some control over the tumor. So it's still in equilibrium phase and what I call the yellow alert phase, okay? The wall of immune suppression is fairly low. So if it's fairly low, yes, by all means, use something to stimulate the immune system to try and break that wall down, or, you know, climb over the wall and not have to deal with it. But anyway, the tumor gets cleared after immune stimulation of BCG, for instance, and then is eliminated. So that's how, you know, initial therapy for, and this would be high risk non-muscle invasive bladder cancer works. So in muscle invasive bladder cancer, you know, notice I've got a little bit taller wall here. And the immune system actually has some control over the tumor. So in this case, equilibrium is headed towards a little bit of that escape phase. Okay, so we have some equilibrium because we have some tumor control. It hasn't gone out to the liver or lungs yet, but it has escaped some because it might be in the lymph nodes surrounding the bladder, definitely inside the bladder wall, a bladder muscle of that bladder wall. So the wall of immunosuppression, I say, is high but not insurmountable. So what do? You can do direct tumor killing, and most people, a lot of people don't talk about this, but I'd like to just mention that chemotherapy also goes after the immune cells, so it can cause immune system reset. Surgery to eliminate the tumor, or trimodal therapy in selected patients, which involves surgery, chemotherapy, and then I meant to put, this should be radiation. Okay, so I put surgery twice, but I meant surgery, chemotherapy, and radiation. And the result here is you can either have elimination of the tumor and then it doesn't come back. Equilibrium for a period of time, which means that, you know, we can't detect it on CT scan, but the immune system's able to control it for a period of time, but eventually it comes back where you have the immune escape again. Or in a minority of patients, rapid escape, and then you have metastatic disease develop, you know, several months after. Now, metastatic disease, this is the Empire State Building, okay? It's tough. When the immune system's lost control over the tumor, you have the escape phase, and that wall of immune suppression, to me, is pretty high. So what do? Well, you don't, in my mind, at this point, there's really no FDA approval where we're actually stimulating the immune system. What we're doing is we need to inhibit the inhibitor, or you can reset the immune system with chemotherapy. But inhibiting the inhibitor means giving immune checkpoint inhibitor. So you're taking the brakes off the immune system again, okay? You are taking that immune system that's inhibitory, and you're inhibiting that inhibitory signal. So 2 minus signs, and you end up with a plus sign, and the patient's tumor gets cleared, hopefully. So hopefully a partial response to return to equilibrium where you have that local control, or a complete response, which is elimination. So how it works, this is adapted from Nature Review Clinical Oncology by Mark Smith, who's also a well-known immunologist in this field of cancer immunoediting. So you have a patient who has a clinically detectable tumor. So that tumor has technically escaped, but it's in some level of equilibrium, right? Because it hasn't gone everywhere, okay? So what do you do? Well, if the immune therapy works, okay, then you either get a shrinkage, so you get more towards the equilibrium phase, more to where the tumor is able to control it better, or if you get a complete response, you're back in this elimination phase again, right? So, and that's good. That's what we want. But if you end up with resistance to the immune therapy, which occurs here, it's going from, you've got the immune therapy here, and then now you're getting resistance. So again, the tumor is always trying to escape, immune control, and then it goes to multiple places, okay? Now, also, you can end up with this in the pre-immunotherapy world. If you weren't getting immune therapy, it would eventually go to multiple places as well. So there's multiple ways that the tumors can escape. It can escape through resistance to the therapy, or it can escape through just overcoming the immune control without therapy. So how does this work? This is from the NCI. It's on one of their websites you can see down here. And I have a couple of definitions here because I really want us to understand how the immune system works. In order to do that, we got to understand some basic definitions. So a T cell is an immune cell that either attacks or doesn't attack the tumor. It depends on the signal sent to it. An antigen is a protein that stimulates an immune response. So it's reported to the T cell. And in this case, with bladder cancer, we're talking about a tumor protein. And again, like I said, antigen, if anyone ever says it to you, simply means something that stimulates an immune response. That's it. PD-1 is a receptor on the T cell. So again, it's a protein on the T cell that's immune inhibitory. So again, you're thinking, okay, we want to block this. And that's why we're giving immune therapies to block it, because you're, again, inhibiting the inhibitor. You're taking the brake off the immune system. And PD-L1 is protein that's on the tumor cell that binds and elicits a T cell immune inhibition. So PD-L1, when PD-L1 and PD-1 bind, you've got that immune inhibition, and then you want to break it, okay? Gets a lot more complicated than this, and I apologize, so I'm not going to get into all the complications, but PD-L1 can also be on immune cells, okay? So we'll talk about that in just a minute. But I want us to understand both aspects, the two primary aspects of how this immune therapy works. You have immune suppression because the PD-L1 on the tumor cell is bound to the PD-L1 on the immune cell. So when that occurs, the protein, this is part of the tumor cell shows up as protein, and this protein is presented to the immune cell. And if it's presented when this is bound, then it's presented in what we call an inhibitory fashion. So that immune system is actually told, don't attack me. Okay, so that's what happens when these two are bound, the PD-L1 and PD-1 are bound, and you have a little bit of tumor cell being shown to the immune cell, okay? And when you block it, you can block it with anti-PD-L1, so you can block it with atezolizumab or durvalumab, or you can block it with anti-PD-1, your pembrolizumab, your nivolumab. And then now that antigen, that protein, that tumor protein, is now being presented in a stimulatory fashion because it doesn't have this inhibitory pathway working. This inhibitory pathway is no longer working because of the infusion that a patient got. So now you've got this presented in a stimulatory fashion. So now you have the immune stimulation occurring. Now the T cell, the immune cell goes and divides, and then bam, goes off and unleashes that immune cell attack. Does it happen all the time? No. Why does it not happen all the time? It doesn't happen all the time because there's a lot more pathways involved than just this. That's one reason why, okay? But, and then this is a little more complicated, but I wanted to also point out a couple of things to those people that already know their immunology and they're saying, Rusty, I'd like to know a little bit more. So I already showed you the tumor and the immune cell interaction, okay? But there's also a third cell involved. That third cell is called antigen presenting cell. You can almost say protein presenting cell, or you can say tumor presenting cell, okay? But that's right here, antigen presenting cell. That's right here, tumor cell. So I've already shown you this. Okay, this is just like the last slide because you have this inhibitory pathway activated, the PD-L1 pathway. This whole thing, when it says peptide, again, all it means is protein. All it means is a tumor is being presented, a little bit of it, to the through this complex. Okay, MHC TCR complex, how the tumor protein is displayed to the T cell to decide whether to attack or not. That's all that means. Okay, so please don't let the jargon complicate things. I know it is a little jargony, but I wanted to show you this because I think it's a nice slide. Okay, and then with immune therapy, again, You get this blockade of this inhibitory signal, you get the activation of the T cell and then tumor cell elimination, okay? Now, what the antigen presenting cell does is it takes a little bit of that tumor cell and then it goes off to the lymph node. Lymph node is the hub of almost the hub of immunology. It's where the protein presenting cell, antigen presenting cell goes and takes a little bit of tumor, or takes a little bit of infection, takes a little bit of virus, whatever, goes to the immune cells, that what we call the T cells, and then it tells the T cells, do we attack or do we not attack? And that decision's made in the lymph node, okay? And then the division occurs, and then you either go and attack or you don't, okay? So in this particular case, the antigen presenting cell or the protein presenting cell, actually, if that inhibitory signal occurs, so again, tumor cell can express inhibitory signal, antigen presenting cell can express inhibitory signal, okay? See, the only thing we're doing is changing the cell type, okay? And then if that bit of tumor there is presented with this inhibitory signal, you get an activation of the T cell and tumor grows, okay? If you give the pembrolizumab or anti-PD-1 antibody or what have you, that blocks that PD-1, PD-L1 signal, then the activation of T cell occurs, and then you can get the tumor to be eliminated because the T cells will divide and then they'll attack. So that's how that works. So there's actually more cells involved, but primarily 3 cells involved: tumor, and tumor can send signals directly to the T cell, the immune cell that decides whether to attack or not, and the tumor saying, "Don't attack me, please don't attack me." The antigen presenting cell is always going around like a security system, picks up that bit of tumor, sends it back to a lymph node, goes to the immune cell, the T cell, and says, "Do we attack or do we not attack?" And that decision is made in part with whether this is activated or not. So a quick primer on a history of immunology and cancer. And I said, AKA, how long it takes for breakthroughs in science, because it really does take a long time, and it takes a lot of people and a lot of effort. But first patient was injected with Coley's toxin, 1893. And then remember, we were talking about Coley's toxins earlier, injecting the infection into a patient to get the sarcomas to be regressed. And then Paul Ehrlich, he didn't have evidence for this, but he proposed that maybe the immune system's clearing the cancer, giving us some protection. And then it took 40 to 50 years. And then now, Mitchison's taking lymph nodes from mice that have tumors, implanting them into mice that didn't have tumors, and then trying to give them tumors, and then finding out, oh, if we take those lymph nodes from one mice to another before we give them tumor, those mice are protected. And that's another finding that shows the immune system can control tumor, okay? Discovery and use of IL-2, that we can use IL-2. It's IL-2, all this is a factor that just induces expansion of these immune cells. And I'll talk about why it's important in just a second. And then, high dose IL-2, again, this factor, if you give high dose in 1985, found that it could actually cause disease regression of cancer in some instances. And then CTLA-4, I know a lot of you probably know that because that's the precursor to the anti-PD-1 therapy that we use, the pembrolizumab, et cetera. That's another protein that's involved in immune suppression, discovered in 1987. And then Steve Rosenberg took the cells out, and immune cells out, and then expanded them, put them back in. That was in 1988 when he did that with his first patients. And then FDA approval of IL-2 for metastatic cancers in 1992, and then PD-1 was discovered in 1992. So PD-1, that protein, that inhibitory protein pathway, in 1992, and then you go all the way up, and I'll show you in a minute how long it takes for FDA approval for us to find it's important in cancer. The FDA approval occurs, treating in patients, all the clinical trials and everything. And then the first time that we took out cells and genetically engineered them, that was in 1989. That's called the CAR T cell, now used to treat patients who have lymphoma. And then Jim Allison, I think we all know who he is, a Nobel laureate, recently won that by discovering and showing that if you block this inhibitory signal, this immune inhibitory signal, CTLA-4 activation, you block it, you can get the tumors to be rejected. I mentioned the first and only cancer vaccines, Sipuleucel T for prostate cancer. Take those immune cells out and then give them factors to help them for inflammation factors to help those immune cells expand. And they also get exposed to a prostate cancer protein. They put them back in the patient. That was developed in 2010. And then that immunoediting that I talked about was, 2011. And then look at this, Look at the FDA approval of PD-1, PD-L1 blockade, 2014. So it's like 20 years after. And so what I, from my experience with working with Indomin 2,3 dioxygenase, which is a pathway involved in tryptophan catabolism, immune suppression, it's late 90s. And then we didn't know that the drug didn't work in melanoma until, the late 2015 to 2019 stage. So 20 years, right? So all of these things, and then finally, those genetically engineered cells are developed in the 1980s, 2017, before we get FDA approval for where they work in liquid tumors. So a lot of things have to happen, and they take a lot of time. So I just wanted to give you that brief overview to show, okay, well, this is how long it typically takes to get from the bench to the bedside, and how much work that actually gets involved and how long it takes. And all the different discoveries that have been made. So for the future, what are we looking at? So for metastatic advanced bladder cancer, you want to look at other ways to inhibit the inhibitor or potentially take the brake off. One of these is LAG-3. It's another checkpoint. Recently, in combination with the anti-PD-1 therapy that's approved for use in bladder cancer, there's a survival difference in advanced melanoma versus anti-PD-1 therapy alone. So could this be combined and used in bladder cancer? Time will tell. Can we modulate or deplete immune inhibitory cells, modulate the immune inhibitory cells to make them immune stimulatory, to make them immune attacking, or just get rid of them wholesale and target them? But in order to target them, we have to better know what they are. And in stimulation, we can maybe do the Steve Rosenberg method, overflow the system with patients' immune cells that have been expanded. For localized bladder cancer, remember, for metastatic or advanced bladder cancer, we talk about inhibiting the inhibitor or maybe depleting or getting rid of these immune cells that are inhibitory. For localized bladder cancer, again, we're talking about immune stimulation. Okay, so maybe sting agonist therapy in the future. What is sting agonist? It's a therapy that stimulates. You remember interferons is coming back again. So these general factors that, stimulate the immune system, stimulate T cell immune cell expansion to try and kill off the cancer and to either get it under better equilibrium or get it in that elimination phase. So quickly, just wanted to go over our development and our working on a model that associates with immune response in bladder cancer. So, I work from the immunology aspect. Kai Aragaki's, David McConkey's graduate students working from the bioinformatics standpoint. David's advisory on the translational bladder cancer science. Noah Hahn, as many of you know, is head of our bladder cancer clinical program to advise on the clinical science aspect. And Jean Hoffman-Sensis, as Dr. McConkey mentioned earlier, she's physician focused on women's health and directs our women's health program. And so we all collaborated on this, and we looked at patients who had these immune cells that respond and attack tumors called T cells. Or you can basically replace this word B cell with antigen presenting cell, or with a cell that's meant to present to that T cell. So you have the presenters, those are the B cells or the antigen presenting cells, the presenters, and then you have the receivers, those are the T cells, those are the ones that receive the signal, and then the two of them decide, hey, are we going to attack or not? And then the signal goes to the T cell, and then the decision is made, do we attack or not? So basically, do we have a tumor where inside the tumor cell you have a lot of these immune cells? And if you do have a lot of these immune cells, you have both the cells that present the tumor, cells that show off the tumor in to the T cell, to the immune cell, and then the T cell that divides and attacks based on that. if you have both of those patients actually associate with better response to immune therapy. And so can we use this in the future to help predict who's going to have the better response to immune therapy? Don't know, but what I can say is it appears that there are some patients who have this particular marker or set of markers that don't have other markers that are associated with the immune response. So it appears it this might be a population that hasn't really been identified previously, but we have to do more research to determine that. Finally, I don't want the goal with the immunology is try to not make it complicated. And then, I'm going to tell you what I just said earlier, that these antigen presenting cells or B cells, the ones that take the little bit of tumor and show it to the T cell to decide whether to attack or not, you need that in order to stimulate an immune response. But now I'm showing you data that shows that maybe these B cells in some instances can facilitate growth. So I want you to know, first of all, that these same cells that I'm saying can be your friend can sometimes be your enemy. And it's hard, and that's why immunology has taken so long for us to find treatments that work, because you have to figure out what signals are your enemy signals and what signals are your signals that you want the immune system to have in order to attack the tumor. So just briefly to show you, this is in mice, and we injected bladder cancer in mice that had an intact immune system. Those are your control mice here. Most of these mice grew tumors. And then a mouse that lacked one aspect of the immune system, and that was the B cell. So it lacked the ability to actually present the tumor via the B cell to the T cell to say, okay, attack me or don't attack me. But in this particular case, you see that maybe the B cells are bad for you in this case, because when you don't have B cells, the tumors are not there. So the point I'm trying to make here is that sometimes B cells are good for you. Sometimes if you have a lot of B cells, that can be good for you, but sometimes B cells can be bad for you too. So these immune cells that we're all saying, can be good for you or bad for you, it's very fluid. And also the immune cells that are bad for you, after you give immune therapy, immune checkpoint inhibitory therapy like pembrolizumab or something like that, they can become your friend again. So it's very fluid, but we're studying this, and the hypothesis from this is that B cells can actually either facilitate tumor elimination or can promote tumor growth depending on the context. And again, I showed you earlier that the signal that is given from that immune cell, the antigen presenting cell, showing the tumor off to that T cell, the cell that can either divide or not divide and attack the tumor or not attack the tumor, okay? That signal between those two cells is critical to determining whether the immune system attacks the cancer or doesn't. And that's what we're trying as immunologists to encourage. We're trying to encourage the immune attack of the tumor. So I just want to thank everyone that's involved with the Johns Hopkins Greenberg Bladder Cancer Institute. Of course, the Greenbergs, thank you so much for y'all's support. I use the word y'all because I'm from Georgia. Dave McConkey, who you just saw, who's our director, Noah Hahn, co-director, director of our bladder cancer clinical operations. And then all these wonderful physician scientists that are here below as well, and also my patients who inspire me. So I want to thank you so much for coming to talk and listening to me.
So thank you, Rusty. So we've had a couple of important questions about inflammation. So earlier in your talk, you were discussing BCG and some other drugs that actually are accelerants, right? They're accelerating the immune system.
Right.
You also mentioned therapy-induced inflammation maybe is kind of a bad thing. So can you help us kind of reconcile those two things?
So there's good inflammation, there's bad inflammation, right? So there's inflammation that can encourage tumor growth via the immune system. And again, it goes back to all those signals that are sent, and the signals are integratory and they're not solo. So that's why the PD-L1, PD-1 axis, the pembrolizumab, the checkpoint immune therapy doesn't work all the time, because There's so many signals being sent, and the signals are dynamic. They're time dependent, which we haven't really talked about a whole lot in immunology, but people are finding out that how long the attachment occurs actually also has an impact as to whether there's a response or not.
Let's maybe pause right there for a minute, because
Yeah.
maybe that's helpful to help understand some of this. So a lot of the time when you get a vaccine, or when you get treated with BCG or some of these other kind of pro-inflammation agents, it's transient.
Yes.
It happens for a little while and then it's gone.
Yes.
Whereas if you're talking about chronic pancreatitis or chronic UTIs,
Yes.
urinary tract infections for bladder, that's sustained.
Yeah.
So this actually leads us then. So in other words, most of the time we're taking drugs to lower inflammation rates, right? We take drugs for pain and all that stuff. When we're stimulating inflammation, we're really trying to do it intentionally and acutely, right?
That's correct. So I think that it's not just, so autoimmune disease, right, is inflammation, but it's the wrong kind of inflammation.
Mhm.
So it's not, I hear people talk about Goldilocks all the time. You want it just right.
Yeah.
It really is about getting the right type of signal sent, the right type of inflammation there, and it being occurred in a way, and going back to your point about UTIs and other things, occurring in a way to where the inflammation resolves
Right.
after eradication of whatever.
Right.
So if you have the chronic UTIs where you have that constant stimulation occurring, well, then yes, you're creating that low level inflammation that is a nidus for cancer. Whereas if you have something where it's cleared quickly, and then it's at the beginning of my talk, I showed the immunity going down quickly after viral clearance. That's what you want. That'll keep the inflammation that is cancer promoting from occurring.
Right. So we've got a follow-up question or statement related to this. Then I want to get into another question about diet. So one of our attendees wonders why BCG causes UTIs.
So BCG, it's actually, I think it's a little more than that. I think the any type of chronic inflammation that's caused can cause UTI. So for instance, bladder cancer itself, you've got a disruption of the lining of the urothelium. And whenever you have any sort of disruption like that, you're gonna cause issues. So, you can have more infections in patients who have those. BCG is a little different. You're introducing a local inflammation, local infection there, and that's a little different from the chronic inflammation that's occurred from the tumor just being there. But yeah, they're both related.
It's again this kind of sweet spot we try to hit where we're stimulating enough inflammation when we're trying to stimulate inflammation without causing it to go chronic. As Rusty said, you want it to resolve at the end when you're finished.
Right.
So one of our other attendees asked, you mentioned diet when discussing chronic inflammation. For someone with non-muscle invasive bladder cancer, any diet recommendations or foods to avoid, for example, sugar?
I get that question a lot. And I actually had that discussion with the patient just last week. They didn't have non-muscle invasive, they had more advanced tumor, but I do talk about that question a lot as a physician. I'm glad, to have this opportunity to have maybe a little more time to expound on that. Tumors are metabolically active. If we have these PET scans you may be aware of where, you put someone in a scanner and they just light up. And the reason is because these tumors are metabolically active. So you like, to increase the uptake of nutrients. But I tell my patients that you have to eat too. The same thing that you eat is just because one feeds the fire doesn't mean that you quit feeding yourself. There's not really what I would say is avoid diabetes. Okay, I would say avoid diabetes. I'd say avoid being obese if you have non-muscle invasive bladder cancer. If you have metastatic, the escape has already occurred, the immune escape I discussed. So basically with me, it's actually getting patients to eat and getting them to really take in whatever nutrients they can get in to keep themselves healthy enough to receive treatment. I tell them at that point, it's about you eating anything you can get in your system to gain the calories, and let the cancer therapy treatment, let us take care of your cancer at that point. So it's two different things. But from a non-muscle invasive standpoint, if you can avoid obesity, you can avoid diabetes, you can avoid all these other health factors, you know, you may not necessarily be protecting yourself from getting your bladder cancer worse, but you'll make yourself have less of a risk factor for pancreatic cancer, for all these other cancers that can get you if you survive non-muscle invasive. So I think just being healthy from that standpoint actually works from an immunologic standpoint as well.
Yeah, and you know, everybody's different. When we talk about precision medicine, it would be great if we could measure the level of inflammation that's in you, and also measure how changing your diet or increasing exercise or perhaps eating more vegetables, things like that, could impact your inflammation. That would be a dream for me, would be able to not only adjust your diet, but actually confirm that it's doing what we want it to do.
Right.
So things like wearables can already measure glucose. That kind of thing could be an important part of our research going forward at some point.
Right.
So Brenda Harrington asks, why would nivolumab control liver mets but allow bladder tumors to continue to grow?
So every patient's different. Okay, so you can get, and the other thing that we're talking about, and actually in GBCI, Noah and David, Dr. Hahn, Dr. McConkey are leading the initiatives on this, is looking at tumor heterogeneity, right? So going back, this immune editing approach is understanding that, to me, helps you understand a lot of things with cancer, because the immune editing that occurs in one cancer, i.e. the liver, might be different than the immune editing that's occurred in the tumor that's still in the bladder, because that bladder tumor hasn't escaped. It's escaped. You got something in your liver, sure, but you still probably have tumor cells in the bladder that are growing, that are under local control, that haven't escaped. Okay, so some of the cells have escaped, some have not. So you basically, in my mind, if you biopsy those two different tumors and you actually looked at different genes that are expressed, you'd probably see some differences. And those differences may be what's impacting the ability of the systemic therapy to control one aspect but not control the other.
I totally agree. I think we would say this word over and over again, heterogeneity.
Mm-hmm.
Some tumors within the same patient look different to the immune system than others, which is frustrating, because you'd think that all of your cancer lesions would respond the same way to a given therapy, but unfortunately that's not the way it works. And we're trying to understand that. So turning now to a few questions that have come in advance. So why is some immunotherapy such as BCG instilled in the bladder, but other immunotherapy is introduced to the body via infusion?
That's a great question. And there's the simple answer, and then there's the more complex answer. And I'm going to try to address both. The simple answer is that you wouldn't want to give an infusion of BCG through the vein to a patient. You'd probably cause them to get sick. It is fine-tuned for local control. So BCG is infused locally because, as I mentioned earlier, it's a stimulatory type of, or accelerator, as David said, type of immune treatment. So it works in the setting of local bladder cancer. It doesn't work in the setting of bladder cancer that's spread to the liver, the lungs, or what have you, because in my mind, because that wall has gotten so high that the immune suppression that's required, that you have to overcome, that immune inhibition that's required to overcome, is so great that stimulators beating your head against the walls no longer go break down that wall. You have to inhibit the inhibitor or take the brake off by using systemic therapy. Now,
now, Rusty, now hold
there's
on.
other reasons. Okay.
Hold on.
Okay.
So you told us earlier that people got high dose IL-2.
That's what I was going to get
And
into.
for bladder cancer, patients got Rofuron, which was recombinant interferon alpha. They got injected with that stuff. Did they feel good after they got injected with that stuff?
No. And see, the thing is, for this high dose IL-2 stuff, number one, you had to admit the patient. Number two, it only worked in a subset. And that again supports the hypothesis that stimulating the immune system once that wall has gotten so high is, and I'm glad you mentioned this, David, this is good. Once that wall is so high, that stimulating the immune system in most cases, now I have to say in most cases, because you got what David just said, the interferon, you've got the IL-2, you know, the metastatic renal cell, you've got the, and then you've got Steve Rosenberg, which has overrun the immune system with all these immune cells that you've expanded in a patient, you've taken out, you reinfuse them back in, and it works, and you get complete responses in a few patients. So yes, stimulating the immune system works in some cases, but the other reason I need to mention is systemic therapy versus localized therapy. So systemic therapy, for instance, in the metastatic disease, you need it to get everywhere. Okay. Now, why they don't do local? This introduces another complication. Why they don't do local PD-1 therapy?
Well, they do.
Well, they do, but
yeah,
but they're
yeah. So Josh Meeks at Northwestern University is actually doing a local intravesical injection of pembrolizumab. Now, I can't comment on the results of that trial because they're not published yet.
Right.
But people are thinking about it. I think what Rusty said about getting it to all the metastatic sites, including those you can't see, is a really good reason to do systemic therapy. And I guess I'm not sure that that stimulating the immune system wouldn't work if it weren't for the toxicity in some cases, as we've talked about.
Yeah, for instance, Steve Rosenberg could probably expand as many T cells as he wanted to put them back into a patient, but you don't want to kill the patient.
Right.
So, and as I mentioned, IL-2, you got to admit the patient. Interferon, I believe you had.
Well, I think they killed a few patients with high dose IL-2.
Yeah.
And interferon makes you feel really sick if you take it.
And the other thing is, going back to your PD-1, you know, intravesical. I mean, it's not FDA approved yet, but I expect, you know, that's a great hypothesis to introduce it locally because of the 1 in 200 death, right? I mean, so the patient's getting it systemically, you know, 1 in 200 will die of pneumonitis, and that's been an argument against using it in the localized setting because most of those patients who have non-muscle invasive will live. So going back to your point, David, local therapy like Josh Meeks is doing is a great idea because you might not get that pneumonitis because it's not going systemic.
Yeah. So let's go down and ask another question that was sent in advance. So can you explain what interferon is and why it is sometimes used as an agent to treat bladder cancer?
So there's actually multiple types of interferon, first of all. So there's interferon alpha, there's interferon beta, there's interferon gamma. So, you know, those are the three primary ones that we talk about. So it's a protein. Basically what people would call it is a cytokine. They're secreted by, you know, the immune system. And there's also these proteins that can receive these cytokines. So interferon, you know, think of it as like I always think of it as like a little triangle just floating around, you know, in the immune system in the body, and then it hits an immune cell that has a receptor for it. It goes in, and then it can cause a bunch of different things. So one thing it can cause is immune stimulation. So it can actually cause expansion of these T cells that I've mentioned before that are very important for the immune therapy working. But And I know, I sound like I sound like a politician because I'm talking out of both ends of my mouth, right? But the reality is that at higher doses, that same interferon, that's another reason why the interferon didn't work in every patient. High doses can actually induce immune inhibitory immune cells. So you have to be careful. So for instance, David and Colin Denny, chair of urology at MD Anderson, they've co-developed this gene therapy that's meant to activate and cause high levels of interferon alpha locally in patients who have BCG refractory non-muscle invasive bladder cancer. And that's great. So in that standpoint, it's actually, the therapy works. It's localized. It's done in a setting where the immune equilibrium is occurring, but not immune escape. And it does, and apparently it's not. There are some patients who don't respond. So you ask the question, okay, well, are those patients, for whatever reason? But it works in that setting. Now, it doesn't work in every setting because of, as I mentioned before, the immune suppression. So it can induce both.
So do you see a day when immunotherapy will eradicate all cancers? And if so, kind of in your crystal ball version of the future, what do you think it's going to take to do that?
So Drew Pardall says yes, and he's been saying yes for 20 years. Drew Pardall is the head of immunology at the Cancer Center. Helped with the initial PD-1 studies in melanoma. His wife is Antigen, who's actually first author on that paper in the New England Journal. So, I would love to say yes. I think we'll get control in a large number of patients. I think hopefully it'll be where we are able to eliminate it in a lot of patients, but I fear that it's going to be like Richard Nixon when he said war on cancer in the 70s. We thought we were going to win in 10 years. And then, we realized we didn't. So there's so many pathways, and the acceleration. So this is another point I wanted to make is back in the day, it was 20 years, from initial discovery to either phase 2 or maybe even FDA approval of an immune related drug. PD-1 showed that to be true. CTLA-4 showed that to be true. IDO didn't work, but that was our experience. But now I think we can get the accelerator on faster. So I think you can get it in clinical trials much faster if you show something working in the preclinical model. Why? Because the community now buys into immunology. The community didn't buy into cancer immunology even 15 years ago, maybe even 10 years ago before CTLA-4. Got FDA approved. So can it happen? Yes. I think we'll get control of immune mediated control in a lot of patients. I don't maybe even a majority, but I don't think it'll be everybody. I think we'll be working on it for the rest of my life.
So I'm optimistic that with all the different approaches that we have and the ones that we're developing, the new measurements that we're developing to see where the cancer is, I think that detecting cancer earlier, there's a lot of progress being made. There may be a day soon where you get a blood test when you go to your doctor for your annual physical, and that tells you whether or not you got a very, very tiny cancer that can be treated with something before it gets into that Empire State Building. I think a combination of all those different things create a pretty bright future for us as cancer researchers and physicians.
I'm excited. There's so many pathways.
I am too. We've got a lot more tools in our toolboxes than we've ever had before, and they're accumulating rapidly. So last call, any final questions? Well, if not, then I want to thank Rusty for a fantastic talk. Oh, here we got one. Oh, thank you so much. Thanks, Brenda. And I'll thank you too, Rusty. Thanks for the talk and the discussion, and thanks to all of you for joining us. Stay safe, and hopefully we'll see you next month. And if you're available, I think Nirmish Singla is going to give you a great talk on kidney health and how it relates to everything we do. Take care, everyone. See you soon.
Bye. Thank y'all.
Bye.